Okay, let me do a very quick introduction to our speaker. I'm very pleased to have my colleague, Dr. Catarina Gratton, speaking at our brown bag today. She's an associate professor in the Department of Psychology. She got her Ph.D. from University of California, Berkeley in 2013. And her research focuses primarily on how to characterize particularly human brain networks in terms of their organization and how they contribute to complex goal-directed behaviors such as focusing or managing attention. And her lab studies the neural substrates underlying these processes in the healthy population, as well as how they vary across individuals and break down with damage. And her team uses a broad range of tools, including fMRI, cognitive tasks, TMS, focusing on what she calls deep precision measures to precisely phenotype brain and behavior in individuals. And the recent work in her lab is focused on individual differences across the adult lifespan and how they're related to preservation and loss of goal-directed behavior with age. And she has a grant that actually focuses precisely on that topic. So let me turn it over to Caterina. Thank you.
Thanks so much for the nice intro. It's great to be here today. I'm just going to go ahead and share my slides. All right. Can you all see the primary slides in front of you? Awesome. Okay, so yeah, so it's great to be here today. I'm relatively new to FSU, so I've particularly been happy to connect through the ISL community, and I hope to do so more in the future. Hopefully this talk will be a little bit of a launching pad. So before coming here this summer, I was at Northwestern University, and we started some of our work looking at large-scale brain networks in the older adult lifespan there, and now we're looking to transition that work to happening here in Tallahassee. So my talk today, so as Neil mentioned, we look at these questions across many different populations, ranging from young adults all the way up through older adults. And the talk today, I will focus more on the older adult side of the project. So it'll be a little bit of a one-off for me, but hopefully it'll give us a chance to sort of touch on topics that we do in the lab, but that might be of interest to those of you in the audience as well. And I thought I'd start off with just a little scenario of a group of friends. Maybe they're all about the same age, but are having different outcomes in life. So let's take Abigail as a starting example. Abigail's doing pretty well. She likes to hang out a lot with her grandkids, but she's also noticing some changes in her cognition, like she sometimes forgets why she walked into a room or where she parked her car when she goes to get groceries. Bob might be having bigger problems in this context, a difficulty with sleep, perhaps some gastrointestinal issues, and he's noticing a tremor in his hand when he's resting and some difficulty making and updating decisions as he goes along. And then Caroline is sharp as a tack. Many folks say she seems just the same as she was 20 years earlier. She exercises daily. She still wins her family game night regularly, and she likes to lead a lot of local clubs. And so obviously this is a wide range of scenarios, and many of us will know folks that fit into each of these different boxes. And our goal as individuals might be to try to make sure we're more on Caroline's path to aging than some of the others that we see here. On average, when we look at the performance of cognitive tasks in folks ranging from the age of 20 over here all the way up to their 80s, we see some common changes that are occurring. For example, some types of functions that we often refer to jointly as fluid cognitive functions, things like long-term memory, groups of executive functions, which help us to achieve goals, working memory, which helps us to maintain representations, these tend to decline across the period of time between the ages of 20 and 80. Whereas other types of functions, for example, related to crystallized intelligence, like vocabulary knowledge or world knowledge, may stay pretty stable or even increase over this time span. But one thing that this plot doesn't represent is where each individual lies relative to this group average change. And so one plot that I like is this study, which was done looking at individuals tracked longitudinally over the ages of 65 to 95 and their performance on a working memory task. So this is a type of executive function task, looking at how we can maintain and manipulate information in mind over short periods of time. And then we can see for each individual, which is plotted as these little line segments, that the general trend is what we see in the group average, that there are declines in working memory function over this time window. And that folks that fit that window, fit that model might be called typical or healthy agers in this window. But you might also notice that there's a lot of other patterns within this plot. For example, some folks are declining much more precipitously, even at early ages. This might be accompanied with different forms of dementia or neurodegenerative disease, whereas other folks are doing just fine, well above average, staying quite stable over these time windows. And these are folks that we might characterize as superagers, people who are doing well in their 70s and just as well as they might have been doing in their 50s. And of course, we might be very interested in trying to push ourselves to one of these groups relative to another, especially in light of the fact that our population is becoming more heavily weighted towards older adults, as shown by this US Census projection of where the population demographics are likely to be in 2060. So it might be important to understand the neural mechanisms which cause us to be in one group versus another group in terms of cognitive function. And this is something that my lab studies. One way that we can look at neural mechanisms of aging is by studying how different regions of the brain respond to or become specialized for different types of functions. But it's also become clear in recent years that many of the complex cognitive functions that we see declines with in aging are actually associated with networks of brain regions that work together to accomplish these different functions. So my lab studies these large-scale brain networks and how they contribute to executive function performance, and we study these issues both in young adults up to older adults. So in the talk today, I'm going to touch on 3 different topics. The first part, I'll just tell you a little bit about how we go about measuring brain networks non-invasively in humans. In the second part of the talk, I'll tell you a little bit about these ideas about precision or deep fMRI scanning that Neil introduced earlier and why we might want to use these approaches. And in the last part of the talk, I'll try to cover a few of our recent projects using these approaches to look at healthy aging and neurodegenerative disease and what's happening in the brain. So I'll go ahead and get started. So my lab uses functional magnetic resonance imaging, or fMRI, to measure brain activity. We can do this non-invasively in healthy humans using an MRI machine, and it allows us to measure brain activity because we can track changes in blood flow, which tends to go to areas where neurons are active and needing more oxygen. And what's really nice about fMRI as a technique is it can give us a picture about brain function across the whole brain at once with relatively good spatial resolution, so we can look at how different brain regions are responding and how they may be interacting with one another. So just to give you a more detailed understanding of this actual measure, what we can do is we can measure brain activity in 2 different regions at once. So for example, here in left and right motor cortex of the brain, and we can measure that fMRI signal over time. And you would see that the signal is very similar across these 2 regions. So if we take a correlation between these 2 time series, you get a very high value. They're very correlated with one another. A region that is part of a different brain system, like a visual region of the brain, would show a very dissimilar pattern with much lower correlations to these motor regions. And I'll just sort of point out that what's been surprising to folks when this was first observed is that this correlation between motor-related regions of the brain is present even when we're asking people to sit very still inside the scanner, not do anything in particular. Often they're looking at a fixation cross, thinking about whatever they'd like. And yet we can see correlations in the spontaneous activity patterns of these brain regions. And these spontaneous correlation patterns actually mimic the patterns of activity we might see in a motor task, like a finger movement task. And so this can be a very powerful approach to map networks of the brain at once across many different brain regions, because we can do it not just for those motor regions I showed you on the previous slide, but many different regions spread throughout the brain. And so I'm going to be showing these to you sometimes in what I'm going to call a correlation matrix, little box I'm showing you over here, where any given cell in this matrix just represents the correlation between a pair of brain regions. And here it looks a little bit like the snow you might have on an old TV set, but that's because I've ordered the regions randomly. If I reorder them such that I put regions in the same network close to one another, you get this really beautiful structure emerge, where you see there are high correlations, these little blocks along the diagonal, and low correlations in the off diagonals. And so this means that there are high correlations among regions that are part of the same network and low correlations between regions that are part of different networks. So we can use community detection algorithms or other types of techniques to identify then the networks of the human brain in a data-driven fashion. And so I've marked them off here with red lines, and I can also paint them on the brain so you can get a more evocative picture of what this looks like over here on the left with different colors. And so this is quite nice. We've now used these spontaneous activity patterns and data-driven techniques to map, networks of the human brain. And we can do this to map low-level networks like the visual network, but also higher-level networks that we think are important for executive functions, such as working memory. And it might seem like a relatively unconstrained technique, but I just want to point out that on average, across large groups of individuals, we can actually produce really robust maps of what these networks tend to look like. So on the left here is the average map across 120 individuals that were scanned at Washington University in St. Louis. And on here on the right is a map of networks from another 1,000 individuals that were scanned at Harvard. They're using different scanners, there's different people within each of these groups, and they're using different techniques to identify the networks, but hopefully you can see, you kind of go back and forth, that they're actually quite similar in which networks they identify and in which locations. And this is true again for both networks for more sensory motor systems like visual and motor related systems, as well as networks that we think of as important for executive functions like the yellow frontal parietal network shown here. And so this one natural question you might ask yourself is how these networks might change over the course of the lifespan, and in particular into older adulthood when we see these declines in many cognitive functions. And so a number of groups, as you might imagine, have looked at this question. And one of the most common findings that you see with brain networks in aging is that these networks tend to desegregate with age, meaning that the networks tend to break down a little bit. The connectivity within the network goes down and the connectivity across different networks goes up. So I can show that to you in these plots over here. This is an average correlation matrix from young adults, those in their middle early years, so 35 to 49, middle late adults, and then older adults shown here. And hopefully you can see, if you can compare across them, that block diagonal structure I was pointing out to you earlier tends to get a little bit more washed out as you go further and further up in age, and you get higher increases in these between network correlations as you go up in age. And so if you calculate a segregation index, how high the correlations are within network relative to between network, you get this systematic relationship where there are declines in the segregation between networks over time. And this segregation tends to be, this loss of segregation, this desegregation, tends to be most prominent for networks involved in executive functions like that frontal parietal network, and it has been shown to correlate with measures of cognitive decline in terms of performance or behavioral measures as well. However, when you look at these images, you'll also notice that there's a fair amount of variability in the system segregation across individuals that were collected in this data set, some folks showing fairly high segregation, even into older adulthood, and again, some folks showing lower levels of segregation earlier on. And so this might naturally sort of beg the question, what's the relationship between this variability we see in cognitive performance with aging and this variability we see in segregation with aging? And so to understand the link between these two, it's really important that we identify very reliable measurements of individual brain networks in individual participants. And this is a topic my colleagues and I have recently been working on quite a lot. And so this brings me to the second part of my talk, looking at precision fMRI and its advantages as well as its applications. I'll take a quick pause here. I don't know, we didn't talk about questions at the beginning, but I'm happy to take any clarification questions on the methods now if folks have them, or I can keep going and we can talk at the end.
I think let's carry on. People, if you want to ask a question, you can always put it in the chat too. I can pass that on to Caterina.
Sounds good. Thank you. All right, so let's talk about precision fMRI. What is it exactly? And so with the typical approach with fMRI, especially these studies of large-scale networks, is to take a relatively small amount of data from each participant. Most studies use 5 or 10 minutes of data, and you pool this amount of data together to create a group average map. And this is very useful. It allows you to overcome sampling error that there is in the original measurement, low SNR in certain regions, other aspects of the noisy fMRI signal, and produce these, robust group average maps that I showed you earlier. However, because you have relatively small amounts of data for each participant, it can lead to fairly noisy measurements for individual subject data. And some of you might be aware, maybe you're not, that there's been a bit of a sort of crisis within the fMRI field over the last few years about the reliability of brain network measurements or brain measurements in general at the individual subject level, with reports such as these that brain fMRI might not be as reliable as once believed. And indeed, there's a number of reports that suggest with 5 to 10 minutes of data, you're at fairly low reliabilities with these fMRI measurements. And quite recently, last year, there was a paper suggesting that with that amount of data, which is typical of very large consortium data sets that have been collected, you're going to need thousands of individuals to produce very reliable brain behavior relationships. And of course, this is a problem if we want to understand which of aspects of brain function are declining with our changes in cognitive function into older adulthood. So our thought is that we needed a different approach. And so the change in approach is actually relatively straightforward. We proposed to collect much more data within a smaller sample of individuals and see to what extent this added amount of data can increase our reliability and our precision of our brain network measurements. We also wed these approaches with additional phenotypic measurements across different states and different sort of information about the individual, and with very good denoising techniques and network definition techniques. And so with this precision fMRI, our hope is to then be able to address issues of reliability that we have with standard fMRI measures, look at the stability of these networks over longer windows of time, which is necessary to understand different types of clinical applications for these techniques, and also how sensitive they are to individual differences in these measures. Okay, so I'm going to introduce 2 data sets that have used precision fMRI in relatively younger adults to explain the advantages of these different techniques. And so the 2 data sets are the MyConnectome project and the Midnight Scan Club project. So the MyConnectome project was a project collected by a professor named Professor Russ Poldrack on himself. He got into the scanner twice a week for over the course of a year. He was at UT Austin at the time. He's now at Stanford. And for each of those measures, he collected some resting state fMRI data so he can get these brain network measures. He also did other surveys and blood draws. So he had a pretty rich phenotypic sampling of his own brain over time. So that's one subject scanned very repeatedly. We have about 80 measurements on this subject once you clean the data up. There's a second data set that was inspired by this initial MyConnectome data set called the Midnight Scan Club data set. It's actually collected at WashU when I was doing my postdoc there. And this includes data from 10 subjects, so more than one, but still not a lot. And each of these 10 participants came in for 10 separate functional MRI sessions. And during each functional MRI session, participants completed these resting state scans as well as scans during different tasks. And I'll just sort of note here, in case any of you are wondering, it's called the Midnight Scan Club project because these scans were all collected at midnight because the WashU scanner was discounted 90%. So it was a project that could be accomplished in a really tight budget for folks. And it was very exciting to be able to get it in. So those are 2 data sets I think have been really revealing about the power of precision fMRI methods. To start with, let's turn to that question of reliability that's been so much in the news, at least in the fMRI world. With the MyConnectome data set, where again, there's this enormous amount of data on a single participant, my colleagues were able to show that you can get actually very reliable measures of an individual person's brain networks. So these are the correlation matrices from the MyConnectome data set and 2 even split halves of the data. And hopefully just again, by eyeballing back and forth, you can see that they're very similar even in the detailed aspects of these correlation matrices. And so if you look at a sort of a test-retest reliability plot, you can see that you can achieve very high test-retest reliability, above 0.95 correlated across the two halves, but it requires substantially more data than has typically been collected in many of these data sets. So here, if you take one half and you set it aside and you say, how much data from the other half do I need to get similar to that first half? You see you need at least over 30 minutes of data to achieve pretty strong reliability to this first half. And this was only one subject, so you might ask, is it something unique to that participant? Well, second, the Midnight Scan Club data set has replicated this result again in another data set, and it's now been replicated in a number of others, that you can achieve high reliability, just requires more data than has typically been collected.
Caterina, there's a quick question there. Do the data sets include people with disabilities, specifically any with ambulatory sight or hearing barriers? Or
yeah,
might that accelerate the loss of cognitive processing?
Yeah, this is a great question. So far, most of the data sets that have been collected of this type have been done in relatively young, healthy adults. These first two are mostly people who are, experimenters themselves or experimenter adjacent friends who came in and were willing to do these types of experiments. I'll talk at the end about some of the projects we have going on in the lab now, which are expanding out to other populations, and a few other groups have been doing that as well to demonstrate that we can do these measurements feasibly in other groups. And I think there's still going to be an open question, do these sort of principles that we have established in this very homogeneous population, to what extent are they true in all of these other populations that we'll measure? And my guess is there will be some differences. I think there will be some factors that are consistent, and I'll try to convince you of that later on, but I would guess that we will see more changes in, say, reliability or stability in these other populations as well.
Thanks.
Alrighty. So the other thing that we looked at beyond just reliability within, say, a scan session is how stable these network measurements can be across different days, or you might ask, as people are thinking about different things or engaging in different cognitive tasks. And we looked at this in the Midnight Scan Club data set, where we again had data from each person across 10 different days. So these are the participants. And then within each day, they completed a number of tasks. There was a noun verb judgment task, a dot coherence task, a motor task where participants were asked to move their hand, foot, or tongue. Oops, sorry. And a categorization task where stimuli were repeated over time and they had to categorize them. And then a resting state condition. And so what we did is we then created, we quantified the similarity in brain networks across these different conditions. So this is another second order matrix, not to be confused with the ones I was showing you earlier, where now any given cell is showing you the similarity between a pair of the connectomes or the pair of the brain networks that we measured from each participant, each task, and each day. And I'll just sort of cut to the chase because there are some other results that I wanted to show you today. But by saying that what we're able to show is that brain networks are actually relatively stable over time and across different conditions. So what you can hopefully see here is that in the background, there's some commonality that are shared by brain networks in all individuals. So there's this sort of group effect. Everybody is somewhat similar to one another. And relative to that, probably the thing that jumps out to you is this really red diagonal we have here, meaning that each person's brain networks look most like themselves, regardless of the day or the task that they're completing. So there are these really prominent individual differences, and they look distinct from other folks that we collect in the data set. There are some effects of task state and day-to-day variability, but they're relatively small compared to these other factors. So we can consider that brain networks are dominated by these sort of stable effects with only relatively minor differences due to these more transient factors. This might make these brain network measurements really good to use in a clinical setting where it won't matter too much sort of how much sleep the person got the previous day, what mood they came in with. It means that we can get relatively stable measurements of their brain networks over time if we get enough data from them coming in. So you might ask yourself, what are these individual differences that we're observing? It looks like each person's brain networks look a little bit different from each other's. So this is a project I did in collaboration with at the time graduate student Ben Seitzman at WashU, where we looked at the locations that were different between each individual and a group average. And so what I'm showing you here on the top is just a continuous map of how similar each location is to a group average map. And you can see that in this subject 6 from the Midnight Scan Club, many locations are this reddish color, quite similar to the group average. And that's the typical pattern. People do look on average kind of similar to one another and fit the characteristics. But you'll notice that there are these locations that are in this blue color that are really quite different from what would be typically seen. So they're less than 0.2 correlated with the group. And so we can focus on these locations to ask what's happening at these locations. Why are they so different from the group average? And what do they tell us about the brain? And these are locations that we call network variants. And so one of the things that we asked, just as a sort of basic measurement question, is how stable are these differences over time? Are they just a factor of, again, measurement error? I told you fMRI can be noisy and unreliable, so it's something we can be concerned about. And what we find is that these locations actually appear again and again across the 10 different sessions in the Midnight Scan Club dataset, leading to very high intra-class correlation coefficients for these measures, above 0.85. So they seem to be quite good measures for identifying these idiosyncratic locations. And if we take all of the locations of these individual differences across people and just overlap them with one another, this is in a sample of 384 other individuals, we see that they're not occurring randomly across the brain, but there's certain characteristic locations where they tend to occur most frequently. And these locations include lateral frontal cortex and near the temporal parietal junction, locations that those of us in the brain imaging world think of as very important for executive fluid functions that we know, for example, decline with aging. In other results from this data set, we're also able to show that these locations of individual differences track with locations that are responding differently during a task that we give them inside the MRI scanner, and that they're related to behavioral differences in performance that we see in tasks we give folks outside of the scanner. So it suggests that these individual differences are meaningful for understanding differences in function that we see across people. So I'll just sort of close this section by pointing out that this high reliability that we can achieve with fMRI with the right approaches, as well as a sensitivity to individual differences, makes these techniques really useful for a number of different applications. So this includes just at a basis understanding differences that we see across normal brains and different pathological conditions, even when there is heterogeneity across these groups. So we can address that heterogeneity. From there, we can then try to create tools that will improve diagnosis and prognosis based on these brain measures. And already a number of groups have demonstrated that these more individualized or precision measures well outperform more classic group average measures in producing prediction of, say, symptoms in PTSD or schizophrenia. These techniques can also be used to then guide different forms of individualized interventions. It could be behavioral interventions or neurostimulation techniques. So, for example, it's been shown that using individual level mapping of brain networks can improve the way that we target transcranial magnetic stimulation in depression. And then a number of groups have also shown that deep brain stimulation efficacy seems to change with the consistency of these brain network results in different locations. So for example, locations that have high consistency across individuals and how their brain networks are laid out lead to highly consistent DBS results. And whereas locations that have low consistency across individuals and how the networks are laid out are the ones that have the most variable treatment responses, as you might expect. And then this, of course, can help us to track treatment efficacy over time and try to understand which cases and what is happening in the brains of folks that are improving with different forms of treatment relative to others. And just one example of how precision fMRI has been used to track the different forms of changes with interventions. There's this pretty striking study in which experimenters took their dominant arm. I think in all three cases, they were right-handed, so they casted their dominant arm, so they couldn't use it for 2 weeks. And they did very extensive scanning before, during, and after this casting of their dominant arm. This is actually a common therapy that's used in different forms of movement disorders. And so what they saw is that there were these really pronounced changes in the functional brain networks for folks when they had this behavioral plasticity to their arm, when they had this casting of their dominant arm, suggesting that these brain network measures can track with these external interventions that are applied. All right, so that hopefully should give you a taste of why you might want to use precision fMRI and what it can add to more standard fMRI techniques. Of course, I haven't yet talked at all about what happens with the aging brain and how we can use it in this context. All of the studies I told you about so far were focused on relatively young adults, and most of them were focused on relatively healthy populations as well. And so in this last part of the talk, I want to tell you a little bit about our work that's trying to extend these ideas into healthy aging and neurodegenerative disease. So as I introduced earlier, one of the most replicated results in the aging literature on brain networks is this observation that brain networks tend to desegregate with age, but that there's maybe some variability in the way this happens. And so this naturally has led us to ask, can precision fMRI be a way to track individual brain networks and understand how their particular networks are desegregating and whether that relates to cognitive decline? But the first question we had to address here was whether or not we can even make these measurements in older adults. Has sort of been addressed. All of these studies have been done in a relatively small sample of participants. So we obtained pilot funding from the Mesalem Center at Northwestern together with NIA in order to conduct a small project in 8 and older adult individuals. And this project was led by my graduate student, Deanna Perez. And so we had 8 folks come in who were between the ages of 65 and 75, and they each came in for 5 separate MRI sessions, as well as 2 behavioral sessions where we did a deeper characterization of their cognitive function. And the goal of this study was really just to establish that we could feasibly collect this amount of data, get folks to come back, and we could get relatively reliable and stable measurements, similar to what we had seen in the young adult population. And so this is just a table of our participant information from this data set. And so one of the things that was somewhat nice from the data set is that we were able to not only collect these 5 sessions from 8 individuals, but 3 of the individuals also had longitudinal data. So we were able to collect another set of 5 sessions at about a year delay from the first. And so, as I mentioned, our first goal was just to establish feasibility to recruit and scan participants for this more extended amount of time. And what I'm happy to report that we were able to do that. And in fact, getting the participants into the scanner wasn't too challenging. Folks were very enthusiastic to do this sort of research. And this was true even though we had the misfortune to start this project in February 2020. So we had a bunch of starts and stops thanks to the pandemic. But the participants were real troopers and we were able to get them in for many of these scans. In addition to being feasible in the sense of getting participants actually to the scanner and collecting the data, as any of you know who have done MRI research or been around it, the real bane of MRI research is movement inside the scanner, head movement in particular. So the other question we were asking ourselves is, could we get high enough quality data where participants were sitting still enough to make our measurements? And this is something we think a lot about in the lab. We try to be very conservative about these issues because they can really bias our measurements. And I'll just point out that we were actually able to achieve very good numbers on these measures, comparable to what we get in our young adult data sets, leading to a very large amount of data on most of the participants that we collected. And so with this large amount of data, we can now look at those same questions I was looking at earlier in terms of reliability and stability in these brain network measures. And so these are the reliability plots that we were able to achieve in our older adults. And so you can see again, time here on the x-axis, and this is retest reliability relative to sort of left out split half of the data. And as you can see, we can achieve pretty high reliability in our older adult data sets above 0.8 with 40 minutes of data, suggesting that we can get very good measurements of their individual brain networks. Of course, we get continued improvements if we go even higher in terms of amount of data. We can then use this data set to ask questions about the stability of these brain networks over time, similar to what we have done in the young adults. And again, we see a similar pattern to what we saw in young adult data sets, where each participant's brain networks, here we have participants on each axis, they look most like themselves and distinct from the brain networks that we get from other individuals. And so this leads to very big difference on the within subject correlations of their brain network measurements relative to their between subject correlations. Now, this was true over sessions collected within a 1 to 2 month period, as we had folks coming in for these 5 different sessions. But as I mentioned earlier, we also had those 3 individuals come back for another set of scans at about a year delay. And we were quite impressed with the pattern that we got, even with this year delay in their brain network measurements. You can see, again, very high consistency within subject relative to across subject, even with this sort of time window, suggesting that there can be quite a bit of stability to these brain network measurements. As I mentioned, part of the goal of this project was to look at these segregation indices. And so Deanna dug into this question within our data set. And we were able to show that segregation indices can indeed achieve high reliability. You can see all the points for a given subject cluster together across the different sessions. And even with this very small sample of subjects, we can recapitulate the pattern that people have typically reported, where older adults tend to have lower levels of segregation than young adults that we've collected in a separate data set. Although you'll also notice that there are exceptions to this rule, which I find very intriguing and something that we want to follow up on. One thing that we've seen, though, with this large amount of data is that the stability of the segregation index measure depends a lot on the amount of data that we're collecting on each participant. And so if you're comparing participants that have, say, 5 minutes of data to 10 minutes of data, you may have very drastically different segregation measures. And it might be important to get this larger amount of data to get a more stable measure of what their true brain networks look like. And finally, we've been interested in tracking those locations of individual differences. I mentioned earlier, the network variance in older adults. And as I said, this is a pilot data set. We just want to establish the how good our measurements are in these older adults. And Deanna's been able to show that with this amount of data, we can get these really robust measures of these individual difference locations. This is in subject LSO3 across 2 split halves of their data. So you can see that we can get very similar locations out of that 2 split halves. So our goals with this project moving forward are to look at these locations of individual differences in more detail to understand what they're representing, if they're telling us something about changes in how the brain responds across different cognitive states, and then looking at that with relationship to a battery of executive function measures that we collect outside of the scanner to see whether or not this relates to differences in executive function that we see in older adulthood. And indeed, we got a supplement to our primary R01 to look at this in a larger sample of 60 older or adult lifespan participants. Is actually going to range from 35 to 75 in order to track these measurements in a similar paradigm. And so this is being led by my research coordinator, Gretchen Wolfekühl. I'm just going to shift gears. This is the last project I'm going to be telling you about. And here I'll tell you about a separate line of work where we've also been interested in looking at brain networks in neurodegenerative disease. And in particular, our primary focus has been on Parkinson's disease, which I'm guessing many of you know is the second most common neurodegenerative disorder, with about 1 million cases in the US and 20 new cases per 100,000 per year. The clinical manifestations of Parkinson's disease are probably best known for their motor manifestations, so things like resting tremor or bradykinesia. But also, along with those motor manifestations, many individuals show pretty prominent cognitive deficits, especially in executive function, as well as often reporting psychiatric issues, sleep disturbances, and other types of deficits. So the clinical and cognitive profile can actually be pretty heterogeneous and widespread, affecting many different domains. The neuropathology of Parkinson's disease is also well known to be associated with these deposition of alpha-synuclein in an ascending fashion, these Lewy bodies. So it starts kind of lower in the brainstem and goes upwards, eventually reaching that cortex at later stages of the disease. And it's particularly well studied in terms of the deficits it causes to the substantia nigra and the striatal dopamine system, and it's thought to be associated very much with the motor deficits of the disorder. But along the way, it also impacts nuclei from many other neurotransmitter systems. One of the best sort of recently studied of these is, for example, the cholinergic system. And both the dopaminergic and the cholinergic system actually have very widespread projections across the cortex. So they can affect many different regions of the brain. So this naturally sort of leads to the question of how Parkinson's disease might affect these distributed networks, and if that can link to some of these other very distributed effects or widespread effects that we see in the disorder. And so this, in an earlier project that I did on this when I was a postdoc at WashU, I worked with my mentor, Steve Peterson, as well as the movement disorders clinic there, Dr. Megan Campbell and Dr. Joel Perlmutter, to look at the average changes that we see in brain networks in Parkinson's disease. And so we looked at this in their data sets, a very well-characterized Parkinson's disease cohort that they'd actually track over time to get many different measurements from them, from cognitive to MRI to PET scans, et cetera, CSF measures. And we looked at the fMRI data in 107 of the Parkinson's participants and 46 healthy controls. These were all non-demented Parkinson's participants and a relatively heterogeneous sample with a wide range of motor and cognitive impairments that's pretty typical of the disorder. And what we ended up finding is a very striking pattern of changes in their functional networks. So this is a correlation matrix comparing Parkinson's to healthy controls. And what you might notice is that there were changes both within networks. So along the diagonal, you can see some of these prominent locations of differences, as well as between different networks and these off diagonals. And this was true in both cortical systems, cortical networks of the brain, as well as subcortical regions. And so one of the locations that we saw differences was in the striatum or the basal ganglia, not surprising given all the past research on this area. But what was maybe surprising to us when we first observed it is that many of the functional connectivity changes that were especially prominent were not associated with the striatum. They actually included motor systems of the cortex, as the somatomotor network, the thalamus, the cerebellum, as well as sensory related systems. And so to us, this suggested that the effects of Parkinson's disease can actually be occurring downstream of the primary locations of pathophysiology in the disorder. And so as I introduced earlier, the real question though is how these might be related to the different clinical manifestations and the heterogeneous pattern of these clinical manifestations in the disorder across cognitive, psychiatric symptoms and sleep disturbances. And so a natural question, especially given my line of research, is to now ask whether or not we can use these precision techniques to reliably map the networks in individual PD participants and use that to understand the deficits that each of these cases is showing. And indeed, we recently received an award from the NINDS to pursue this research and with my co-PI, Megan Campbell. She's a primary site PI at WashU. In which we're going to collect precision fMRI data on 70 individuals with Parkinson's disease, as well as 50 age-matched controls in order to track their individualized network patterns. Alrighty, so that gives you a little bit of a taste of some of the work that we're doing in the lab. So in the talk today, just to summarize, I showed you a little bit about how do we measure brain networks in general in humans and why we might want to do this in a precision fashion with this really extended amount of data. And then the last part of the talk, I introduced some of the work that we're doing in the lab looking at precision fMRI and healthy aging and neurodegenerative disease. And so hopefully this work suggests to you that this might be a real avenue where we can start to investigate differences that we see across older adults and their differences in their brain networks and how they may be related to one another. And hopefully this can help with improvements to treating the condition in the future. And so I'll just close by thanking my collaborators on these projects. I've particularly highlighted the folks that have been doing the aging and Parkinson's disease work here. I also want to put up my contact information in case you have any questions that you don't have a chance to ask at the end of this, although I'm happy to take questions in a second. And I'll also point out that we are interested in recruiting participants here in Tallahassee between the ages of 18 to 75 who might be interested in coming in for many repeated MRI sessions. And so I have our contact information and phone number in case you or anyone you know might be interested in participating in this work. And thank you all for listening to the presentation.
Thanks so much for an excellent presentation, Caterina. And we do have some time left, thankfully. You timed things beautifully. So we have a lot of time for questions and thoughts, comments, or yeah. If people want to just, I guess, unmute, you can ask the question personally, or just put it in the chat and I'll ask it for you. All right, well, why don't I lead off on that?
I think Dawn has her hand up.
Oh, does Dawn? Oh yeah, I see you've got a hand up. Thank you, Dawn. It's hard to see on my screen.
Sorry, I wasn't sure if you wanted me to jump in or not.
Absolutely, please do.
Sorry. Yeah, so first of all, that was totally fascinating in lots of ways and really exciting work. I have questions, or probably things you can't answer exactly, but I guess something that's sort of on my mind. I'm thinking about the work of people like Denise Parks, who have talked a lot about sort of life course exposures and how people develop what I assume for my less sophisticated understanding of these things, patterns in how they use their brain, and that changes to some degree over time, but it's related to kind of life course exposures. But probably at some point there's sort of consistency, I would assume, where there these differences, these pattern differences that you observe become more set. And we'll just say they have these sort of unique ways. So I think about two interesting aspects of that, which is there might be certain ways that people use their brains that vary, especially in a world where we're more cognizant of neurodiversity. And so there might be sort of groups early on in the spectrum that figure out effective ways to leverage their brain given variation, normal and sort of out of the norm variations in brain use. But then not just those variations, but maybe some pattern differences that might then go on to be predictive way early on, before we see someone who's already a Parkinson's patient, of like, wow, people with this combination of patterns of brain use do you think there's a possibility that someday we could get to that place? So way before people have dementia or symptoms or any of these things, that there might be a chance to sort of say, oh, with these brain images, we might get to a place of sort of early diagnosis, I guess.
Yeah, those are a great set of thoughts, and I'll try to address all of them, but definitely bring me back on track if I'm missing some pieces. So definitely, I think this idea that we might sort of, our brain networks might get stabilized by repeatedly engaging in certain activities or having certain environmental exposures or sort of learning exposures is definitely the way that we think about it as well. And I think there's, some evidence to that fact that these brain networks do change with sort of repeated training and particular exercises over shorter time windows. And you can only imagine that that'll be magnified if that's over a full lifespan, or especially if it tends to happen at critical periods. And it's something that a lot of folks are interested in investigating, and we are too. One very basic question, for example, in that domain that we have with our data set is if we're going to see bigger individual differences in our older adults than we get in young adults, because there's been more time for this sort of accumulated experience to have its effect on these brain networks. And so I think there's a lot of very basic sort of questions like that to answer, and that'll really be really important. We've done a little bit of work in the young adults showing that these individual differences in brain networks are affected by genetics. So if you take twin samples, they'll tend to be more similar to one another than, say, identical twins are more similar to non-identical twins, and then unrelated individuals. But that's certainly only a piece of the puzzle. So I think there's a lot to be done there. And then I guess in terms of thinking about these questions in terms of prognosis and if they can be used in order to improve our sort of future understanding of folks who will have dementia, or which treatments might be better for some folks relative to others, I certainly hope that's true. I don't think we're that close to that point, honestly, at the moment. The truth of the matter is we need a lot more longitudinal data in the field. A lot of the data that exists is cross-sectional, including a lot of the data sets that we're looking at. At most, we have longitudinal data maybe from 1 to 3 years or so, which isn't really enough to get that picture of like someone early on when you'd want to intervene. And so I think that is going to change. There's more of a push to having more and more longitudinal data across data sets. There are, for example, some Alzheimer's data sets that are starting to get longitudinal data. The Parkinson's data set from my collaborators at WashU, they do get longitudinal measurements on these folks every 3 years until death, actually, for most of the folks. And so they're able to track them over time. And I think that's really going to improve our understanding of the trajectories within a given person and what we can do in those cases. We do have one data set within the lab where we have some data across a span of about 14 years, just on a small sample of participants, but it already sort of gives us a hint of whether we could even answer these questions with brain imaging data. I will say that there is some data from the Alzheimer's disease world, though, that suggests that these brain network measurements, or these sort of fMRI measurements, are one of the earliest ones to show changes in folks that will later develop dementia. So I think it's promising to have these as candidates to continue to investigate.
Thank you.
Other questions? Caterina, can you maybe stop your screen share and we can,
Of course.
I can patch if anybody's got a hand up because I can't see it otherwise. Let me ask a couple of follow-up questions to kind of Dawn's line of inquiry. So we know particularly that brains tend to specialize, as you point out, when people engage in a lot of activity that's highly focused. For instance, experts, and the example that comes to mind is the amount of cortex that somatosensory cortex that are taken up in, say, left and right hands. The differential for people who are string players, where their right hand doesn't have as much cortex, somatosensory cortex devoted to the motor side as do the finer fingering that you would see with a violinist, something of that sort. And so we know that as people develop expertise, their brains tend to specialize, and maybe they even have specialized networks. There's some hint of that, particularly in music, that on tasks that you're looking at fMRI, that musicians might process some of these tasks quite differently in literally in different hemispheres. And so I guess my question is, some of the consistent but highly high individual differences in cortical areas, could they be representing in part that type of process?
Yeah, I think that's certainly the case. We haven't looked at it consistently from the point of view of, people who are very specialized for one function, where we could go after that question very exactly. But it is certainly something, it's how we think about it, and one of the projects we'd be really interested in developing in the lab. There's an interesting result from recently from Ed Fedorenko's group at MIT, who's been studying this question in the domain of language, where she's been for a while looking at these sort of very precision, individualized ways of mapping language in the brain, and has recently conducted a study on polyglots, so folks that know many different languages. And what's interesting about the results of that study is that she's able to show that those individuals actually have a very similar language network to what we get among other folks, which every person differs a bit, in where they are exactly, but they have the same major components. But their language networks are actually smaller than most other people's, is the sort of the main result that they're finding, which is very counterintuitive. At least to me, it wasn't what I would have expected. I don't think it's what they were expecting as well. So I think it tells us that there's more to learn about how, training and how expertise is actually going to interact with these variables about organization and specialization within the brain.
Actually, I would have predicted that based on some of the early work on chess, showing in fact that smaller, when you give a chess task to an expert as opposed to someone more novice, that the expert in fact activates far smaller amounts of cortex in the processing or the solution of that problem. So it really looks like you get these very high precision circuitry developing to enable people who are highly skilled to do their tasks. So they don't in fact take up a lot of extraneous effort or in this case, I guess, energy expenditure in the cortex to accomplish the same tasks. So yeah, that one fits with my intuition. Let me ask another individual difference question. Male, female. Male brains tend sometimes to be specialized a little differently. Some of the early work, I don't know the current work particularly for language representation, but it tends to be a little bit more bilaterally represented, cortical processing for language tasks in women than men, and women tend to recover better from strokes. People have hypothesized in part because of that fact that they have more distributed representations. And so my question was to kind of blend those two things, aging and sex or gender in this case, and ask the question of those patterns that you observe of dedifferentiation in the cortex with age. Do those patterns differ between men and women, or are the samples not quite large enough to break it that way?
Yeah, that's a great question. So I guess the first order answer is that yes, there are going to be differences between men and women and male and female brains. They're not super straightforward. They're not necessarily as big as some of the other differences that we see. And there's obviously a lot of variability and overlap across the groups. We haven't looked at it specifically in the context of the segregation measures. And our sample's small, so I wouldn't really trust it if we did. But the original result that I was reporting from the WIG study is actually Denise Park's data set. So that one would be large enough to look at that. And I can't remember them reporting a difference, but we should ask them. I bet they have looked at that. Obviously, we've had a little bit of an issue in lab. I don't know if you have similar issues where we also tend to have a lot more women participating in our studies, and it's something that we have to be a little bit cautious of as we're doing recruitment. So especially if any folks have good advice for getting those men into the lab, we would really appreciate the help on that front. And that'll give us
it's a survival effect, unfortunately, for us males. We don't live as long as females do.
We have the issue in the college age students sometimes too, though, so I don't know what it is.
That's a common problem, I understand it, but for different reasons at different parts of the lifespan. The reason I asked that de-differentiation question is that if we think about abnormal aging, particularly neurodegenerative disease processes, for Alzheimer's, one of the biggest risk factors is being female compared to male. And so that's why I was wondering about the normal course, if we know what that is, for the de-differentiation pattern or lack of integration pattern in women versus men, because maybe that's a lever of sorts to get at the differential prevalence of Alzheimer's disease in women versus men. Part of it is, of course, women live longer, and so they have more of an opportunity for their brains to degenerate or be struck by a degenerative disease. So I think that's a piece of the puzzle, but it seems to me like the sort of precision mapping that you're doing could give us an interesting lever into understanding some of those differences. And that de-differentiation measure, which seems to be reasonably stable, could be a really nice biomarker for normal versus abnormal aging.
Yeah, I think we're definitely on the same page. Sorry, my computer keeps trying to update. Hopefully it doesn't happen during this. Yeah, I think we're totally on the same page. And I think that's a really interesting question to look at. I've mostly focused on Parkinson's disease, partly because of my interest in executive function and sort of past collaborations. But in some of the work that we were doing, especially when we were at WashU, was also comparing the changes that you see in brain networks in Parkinson's disease compared to Alzheimer's disease. And also at WashU, they had this really fascinating sad data set of folks that have dominantly inherited Alzheimer's disease, and they were scanning them over time. So that sort of gets back to that longitudinal question and sort of predicting early markers of change there. And what's interesting is that all of those conditions show changes in brain networks, but they're also the network patterns were a bit different. And interestingly, maybe more similar in some ways between the Parkinson's group, the idiopathic Parkinson's and Alzheimer's, than the dominantly inherited Alzheimer's and the idiopathic. So it suggests, there's going to be different, as many folks have been talking about, different sources for these disorders. And we need to be careful about sort of distinguishing across these different forms of heterogeneity. And maybe sex is another way to go after that and getting these samples where we can get different types of biomarkers in each case.
Appreciate that. I've been dominating here, and we're just about out of time. Other questions for our speaker?
And that's a great question. Thank you.
Jump in, Juan.
Yeah, thanks for the great talk. So I was wondering, have you looked at how sensory decline, how that would change the brain network pattern, and then after you adjust the sensory decline, for example, with hearing aid or maybe other assisted device, how that changes the brain network again, just thinking along that line.
Yeah, we have not directly looked at that in any of our data. I think it's a really interesting question. We've looked, so I think some of the work that's being done in the young adults maybe speaks to this. So for example, not the folks that had their arm casted for a period of time, so they just didn't have access to their dominant arm for use, had this really prominent plasticity in their brain networks within the course of about 48 hours. They were seeing changes where, two of the most strongly connected regions of your brain are usually left and right motor cortex. That's why I used it as the first example. They show really high correlations. You're going to be able to see that in any data set. And those went from something like 0.8 correlates, which is what you typically see, it to like 0 in some of those participants when they had their arm casted. So it suggests that these changes in the way that you're interacting with the environment are going to have and can have these really prominent effects on your brain networks. Interestingly, they went back to normal after the cast was removed. So it's a little bit of a question of how long that change will last and if there's different ways of affecting that change. But it's a really interesting question. There's another group that we're collaborating with that has also scanned folks that are congenitally blind, and they're interested in tracking how our, say, visual networks changed in folks who are congenitally blind. And again, you see some patterns that, must be sort of in place, and they're similar to what you see in group averages who are normally sighted, but then you also see patterns that are very different in individuals who are congenitally blind. So I think that'll be a really interesting avenue to look at.
Thank you. Anybody
else? If not, thank you so much, Caterina. Very much appreciated. Wonderful talk. And one of the advantages to having these presentations is hopefully other people will contact you who've got interests that align with some of yours. Thanks again. Yeah, please be in touch. Thanks for having me. It was really nice to be able to talk to all of you.